Alright, in this video, I'm going to explain another coordinated strategy to for cancer prevention. And this one's going to include a grapeseed extract, which is a really powerful supplement or extract that you should include. So basically, the first step is a normal HSP70 GP96 anti-tumor pathway, which is what tumors try to block. Under immunogenic stress, HSP70 and GP96 bind tumor antigens. They traffic to the tumor cell surface with calreticulin, which is CRT in short. The CRT acts as an eat me signal. The CRT can actually hide in the tumor, but the goal is to bring it out to the surface, because if it's out on the surface, then the CD91 on the macrophage and dendritic cells can bind to the CRT, and the result is efficient phagocytosis, which is the engulfing of the cancer cell. This also happens to be one of the strongest bridges between innate and adaptive immunity. Okay, so a second step is a tumor blockade strategy, which is downgrading the CRT via lipid oxidation. So tumors increase lipid oxidation via oxidized LDL. Oxidized lipids disrupt ER stress signaling and impairs CRT, short for calreticulin, translocation to the cell surface, which means less CRT, and which cannot effectively dock antigens for CD91 uptake on the macrophage. So the tumor becomes invisible despite antigen presence. That's what happens when the CRT is not shown on the surface of the cell and is instead hiding. So the third step is a CD47 SERP-A dominance, which is don't eat me. So tumors upregulate CD47. As a result, the CD47 binds to the SERP-A on the macrophage, because the macrophage has or expresses SERP-A. So this overrides the CRT eat me signaling and actively inhibits the phagocytosis, even if CRT is present. Another way tumors avoid immune surveillance is the suppression of CD91 on macrophages. So the tumor microenvironment with high oxidized lipids and iron-loaded macrophages, or in other words, iron trapped in macrophages, this actually reduces mitochondrial ATP, which means weaker mitochondria, weaker mitochondria, which means weaker immune cells, because immune cells need ATP. So this leads to a downregulation of CD91 expression, less antigen cross-presentation, because the CD91 has to be expressed on the macrophage so that the CD91 can bind to the calreticulin on the cancer cell. But that can't happen if it's downregulated because of high oxidized lipids such as oxidized LDL. Now, a way to reduce oxidized LDL is to eat foods like oranges. Oranges can reduce oxidized LDL. There are other ways to reduce oxidized LDL, like a red cabbage, possibly other fruit. So your diet is important here. And another way cancers avoid immune evasion or immune surveillance is altered lipid oxidation through epigenetic silencing. So basically, an oxidative lipid stress that closes chromatin at immune genes. So basically, the chromatin has the DNA inside of it. If the chromatin is closed, then the immune genes for the DNA cannot be activated, which means suppresses transcription of CD91, MHC12, and co-stimulatory molecules, CD80 and 86. Or in other words, that the immune system is weaker. It can't attack cancer cells. Okay, now where grapeseed extract fits in, or GSE for short, is grapeseed extract is rich in proanthocyanidins, which act at multiple nodes of this blockade. And so grapeseed extract, like I said earlier, lowers oxidized LDL, which means that a chromatin can stay opened. If the chromatin is opened, then the genes and transcription factors for the immune system will work. Grapeseed extract also scavenges lipid radicals and preserves plasma membrane trafficking. This restores CRT surface exposure. So basically that means that the grapeseed extract is bringing the calreticulin in the cancer cell out to the surface instead of the calreticulin hiding in the cancer cell. So which means that the CD91 on the macrophage can bind onto the CRT on the cancer cell's surface and take out the cancer cell, which with engulfing or phagocytosis. So grapeseed extract also stabilizes HSP70, GP96 CRT complexes, which is what cancer tumors try to escape. So the polyphenols in grapeseed extract protect protein conformation, reduce oxidative unfolding of chaperones, and supports antigen chaperone integrity, or in other words, antigen presentation. Antigen presentation means that the immune cells can see the cancer cells. So this improves immunogenic cargo delivery. Grapeseed extract also relieves the CD91 suppression on macrophages because the CD91 has to be showing or expressing. And it does this by reduced lipid oxidation, improves mitochondrial ATP, reserves iron-induced metabolic shutdown. As a result of grapeseed extract doing all of this, this favors an upregulation of CD91 expression on the macrophage, which upregulates phagocytosis and cross-presentation. This also reopens the HSP70 GP96 CD91 axis. This also grapeseed extract also stops the tumor from expressing its CD47, which binds onto the SIRP alpha or SIRP alpha on the cancer cell. Because if the CD47 binds to the SIRP-A, then the immune cell can't do phagocytosis. So grapeseed extract lowers oxidative stress, driven CD47 upregulation, which means that the CD47 on the cancer cell can't be upregulated. Grapeseed extract shifts macrophages away from SIRP alpha dominant inhibitory signaling. This means that it does not block CD47 directly, but weakens its dominance. So calreticulin signaling becomes competitive again. So basically that means that grapeseed extract lowers the SIRP alpha on the immune cell so that the CD47 on the cancer cell can't block the SIRP-A, which would stop phagocytosis or the engulfment of the cancer cell. Also, grapeseed extract is an epigenetic reopener because the proanthocyanidins inhibits histone deacetylase activity, because if histone deacetylase was active, then that would close the chromatin, and if the chromatin is closed, the genes for the DNA inside of the chromatin won't be able to work. So this restores antigen presentation machinery through grapeseed extract support of NRF2 dependent antioxidant gene expression, which would also not be able to be expressed if the chromatin was closed. So the outcome of all of this is, and it's also the recap for the whole video, is grapeseed extract upregulates calreticulin, or CRT in short, on tumor surface, upregulates functional HSP70 GP96 antigen chaperoning, which is what tumors try to escape, upregulates CD91 expression and signaling on macrophages, which is what macrophages need to bind to the CRT on the cancer cell. Grapeseed extract also reduces lipid oxidation-driven immune paralysis and reduces effective dominance of CD47 SIRP alpha, which means that the CD47 on the cancer cell binds to the SIRP alpha on the immune cell, which means that the immune cell cannot do phagocytosis on the cancer cell. So there's a lot more to fit into this, but the video would be way too long. Subscribe, stay tuned for more videos, and I'll see you in the next one.